Julia Rohrberg. I'm a Susan G. Komen Postdoctoral Fellow, and I'm working in the laboratory of Dr. Andrei Goga at the University of California in San Francisco. I did my PhD in biochemistry and cell biology, so I was trained as a basic biologist. In my postdoc here at UCSF, I'm studying the cell biology of triple-negative breast cancer. During this webinar, I would like to give you an insight into how we can study triple-negative breast cancer in the lab. Understanding triple-negative breast cancer on a molecular level is very important to eventually find a way to treat this aggressive cancer. Let me give you an overview of what this webinar will be about. To understand what we are working on in the lab, I have to explain to you what cell division is, because cell division is fundamental for the growth of cancer and will make you understand what cancer actually is. I will then talk about breast cancer and the different subtypes of it. I will go into treatment options for the different subtypes and briefly explain how they work. I will then give you an insight into our work in the lab. We found a factor called MYC to be critical for the survival of triple-negative breast cancer. I will explain what MYC is and how we can take advantage of the fact that MYC is high in triple-negative breast cancer. And at the end of my presentation, I will introduce the best method we have to date to test potential new drugs in the lab before we test them in patients, the patient-derived xenografts. Everybody of us started out as a single cell, a zygote made from the fusion of our dad's sperm and our mom's egg. And we are growing or getting bigger, not because the cell is getting bigger, but because cells divide. That means cells make more cells. The human body is made out of 37 trillion cells forming around 200 different tissues. Cells from different tissues have different functions, such as transporting oxygen, communicating between different parts of the body, forming a barrier to the outside, or defending us against pathogens. And many of these cells replace themselves, but some cells never divide. Cell division is a carefully controlled process. It can be turned on when needed and turned off when not. An example is the wound healing process. An injury stimulates the cells to divide rapidly. As the injury heals, cells come into closer contact, rate of cell division slows. And when cells touch each other, the division stops. So a cell can receive signals from inside the cell. For example, if it's injured, it will stop dividing. It can also receive signals from outside. For example, growth factors and hormones will tell the cell to divide. However, if the cell touches another cell, it receives the signal stop dividing. So a normal cell will stop dividing if it receives a signal from outside or if it realizes it is damaged and cannot function properly anymore. In that case, it can even undergo cell suicide and die. In cancer, however, the cells don't respond to the signals anymore. They divide even if the cell receives a signal from outside to stop growing or if it's damaged. This leads to many uncontrolled cell divisions and the formation of a tumor mass. So cancer is basically the loss of normal growth control. In case of breast cancer, this uncontrolled growth happens in the ducts. Let me briefly explain the anatomy of a female breast. Each breast has 15 to 20 sections called lobes arranged like the petals of a daisy. Inside each lobe are many smaller structures called lobules. And at the end of each lobule are tiny sacs, the bulbs, that can produce milk. Lobes and lobules are connected by a network of thin tubes called ducts. The ducts transport the milk to the nipple. 80% of all breast cancer happens in these ducts, which is then called ductal carcinoma in situ. Carcinoma refers to any cancer that begins in the skin or other tissue that cover internal organs, and in situ means in its original place. Some cells can start to break out of the inside of the milk duct. This is what is then called invasive ductal carcinoma. These cancer cells can then enter the bloodstream or lymphatic system and travel from the breast to other parts of the body. There, the breast cancer cells may again begin to divide too quickly and make new tumors. This is called metastatic breast cancer. Commonly, metastatic breast cancer arises months or years after a person has completed treatment in the brain, lung, liver, or bone. So why are metastases a problem? Because in the liver, lung, or brain, a big tumor can prevent the organ from functioning normally and can even cause death. How can we treat breast cancer? To answer this question, clinicians have to determine which breast cancer the patient has. Breast cancer is not a single disease, but is divided into several subtypes. In the clinic, the expression of 3 cell surface receptors are determined, which are ER, PR, HER2. Each subtype can be profiled much more complex using molecular and genetic information from tumor cells. These molecular subtypes are then called luminal A and B, HER2 positive, and basal-like. However, at this time, these molecular subtypes are used mostly in research settings and are not included in pathology reports. I'm going to explain to you now what are ER, PR, and HER2. ER, PR, and HER2 are so-called receptors. ER stands for estrogen receptor, PR for progesterone receptor, and ER, sometimes also called ERB2, stands for epithelial growth factor receptor. Receptors can be found on the surface of a cell. You can imagine these receptors as the eyes and ears of a cell. I was telling you before that a cell can receive signals from outside the cell, and they are the guys receiving the signals and telling the cell what to do. And these signals are hormones which circulate in the bloodstream. If the right hormone comes along that fits into the receptor, like a key fitting into a lock, the switch is turned on and the cell starts to grow and divide. However, if the cells are not normal but cancer cells, these hormones can fuel the growth of a tumor. If breast cancer has these receptors, it's called receptor-positive breast cancer. You would first think it's bad because the growth of the cancer is accelerated by the hormones, but we can take advantage of the presence of the receptors. Hormonal therapy can be used to interrupt the influence of hormones on the growth of the cells. The drugs can prevent the hormone from binding to the receptor, and so the cancer cell doesn't receive the signal to grow anymore and might even die. I list the drugs which are used for ER/PR positive breast cancer and HER2 positive breast cancer. If a patient is diagnosed with triple negative breast cancer, that means that the cells of the tumor don't have any of the receptors. And that means that the cells of that tumor can grow and divide even in the absence of hormones and receptors. And that also means that the drug cannot block the growth of a triple negative tumor and are ineffective. 10 to 14% of breast cancer is triple negative. This subtype is the clinically most challenging, as it is the most aggressive form of breast cancer with the poorest outcome and the highest metastatic potential. It is diagnosed more frequently in younger women with BRCA1 mutation, and some studies have suggested that triple negative breast cancer incidence is higher in some African American and Hispanic population women. No targeted therapy exists. Chemotherapy is the standard therapy. Only 2 approaches have been proven efficient against triple negative breast cancer, and that are the anthracyclines and the taxanes. There are also other strategies tested in the clinic, but we will have to see which ones will be beneficial for the patients. I want to explain to you now how taxanes work, because this is important for the research I'm doing. So for understanding how taxanes work, I will have to tell you a little bit more about cell division. I already told you that cell division is an important process for the tumor to form. But as you can imagine, this comic I drew is very simplistic. Let's dig a little deeper into a process called mitosis. Each cell contains DNA, which is genetic information each of us carries and which we get inherited from our parents. With each cell division, the cell has to make sure to distribute this genetic information evenly between the two daughter cells. The cell is doing that with the help of something called microtubules. You can imagine them like strings grasping onto the DNA to pull the DNA apart so that the cell can divide into two cells. The structure the microtubules are forming is called mitotic spindle, and the whole process of cell division is called mitosis. And we can visualize that process in the lab. These are pictures of cells I took with a microscope, and we can stain the microtubules green and the DNA in blue. And we can even see the sides where the microtubules grasp onto the DNA in pink. During chemotherapy with taxanes, these microtubules get destroyed, and so the mitotic spindle cannot form, and the cancer cell cannot divide, and the tumor shrinks. And because the taxanes are destroying microtubules, they are also called microtubule targeting agents. And taxanes, as I said, are part of the standard therapy against triple-negative breast cancer. However, I pointed out at the beginning of my talk, a lot of other healthy cells in our body are dividing as well. And they do that using the same process called mitosis. And unfortunately, chemotherapy does not know the difference between cancer cells and normal cells. So also healthy cells that divide will be killed by chemotherapy. So now you can imagine why there are side effects of chemotherapy. The normal cells most commonly affected are the blood cells, the cells in the mouth, stomach and bowel, and the hair follicles, which results in low blood counts, mouth sores, nausea, diarrhea, and or hair loss. So you see that we cannot use these chemotherapeutic agents indefinitely because the side effects would become too severe. And this is the reason that sometimes the tumor is not killed entirely and some cells are left over, which then begin to grow again when chemotherapy stops. So there is an urgent need to find new and better therapeutic against triple-negative breast cancer. And to be able to do that, we have to first deepen our understanding of triple-negative breast cancer on a molecular level. We have to know what is going wrong in these cells to find something that we can target. And ideally, this would be something that is different from normal cells so that we can interfere with the growth of cancer cells while sparing the normal cells. And so there is a big effort to understand breast cancer better on a molecular level. For that, the I-SPY trial was initiated to identify diagnostic markers, test hypotheses, and develop new treatment strategies against breast cancer. The patients in this trial were treated with standard chemotherapy and surgery, and biopsies of the tumors were taken and extensively characterized. And so a former colleague of mine took the data from patients with triple-negative breast cancer and asked the question, what is different in these tumors? He found that a protein called MYC is significantly elevated in triple-negative tumors. So these tumors, as we know, don't have ER, don't have PR, and don't have HER2, but now we know that they have a lot of MYC. Patients with tumors that had very low levels of MYC, shown here in blue, had much longer disease-free survival than patients Most tumors had a medium amount of MYC shown in pink. Patients with tumors that had the highest amount of MYC, shown here in orange, had the shortest disease-free survival of all patients. Also, increased MYC was associated with early disease recurrence. That means patients with tumors came back early after chemotherapy had the highest level of MYC. In the lab, we got more direct evidence that MYC indeed plays a role in the development of breast cancer. We actually can introduce MYC into the breast tissue of mice. And these mice will rapidly form breast cancer. And interestingly, when we take away MYC, the tumor disappears. This shows that MYC is indeed important for the development of breast cancer. And the anatomy of the murine breast is very similar to that of humans. So we have a very useful tool in hand to study breast cancer in the lab. We know now that this protein called MYC seems to play a very important role in breast cancer. But what is MYC? MYC is involved in regulating many processes in the cell, like the ones we already talked about, cell growth and division and cell death. But it is also important for many other processes like the immune system or blood vessel formation and many more. And these are all processes that are very important for a tumor to survive and to grow. MYC is not only found in breast cancer, but in many other cancers. It is in fact the most frequently amplified oncogene in human cancers. An oncogene is a gene that transforms a normal cell into a tumor cell. MYC overexpression occurs in 30% of all human cancers and frequently predicts for poor clinical outcome, aggressive biological behavior, increased likelihood of relapse, and advanced stage of disease. And so therapeutics against MYC could potentially be used against a wide range of human cancers. However, nobody was able yet to find or develop a drug that efficiently inhibits MYC. That's why MYC has been deemed to be undruggable. But can we maybe target the interactions or pathways that are important for MYC to fulfill its function? And that's what we are trying in the lab. And several years of work in the Goga lab actually revealed that indeed, if we inhibit cell division, cells that have a lot of MYC will die. So now I want to show you a little bit how it looks when we work in the lab and how we can study cancer outside of the human body. So that's me working with breast cancer cell lines and looking at them under the microscope, and that's how they look. Breast cancer cell lines are cells that came directly from a patient's tumor. The cells were taken from a biopsy and plated into a plastic dish that is treated so that the cells can grow. And these cells will grow indefinitely, so we can expand them to do experiments with them. And there are multiple cell lines available, from receptor-positive breast cancer as well as triple-negative breast cancer, and also normal cells that are not cancer cells. And so we took these cells and we were trying to find something that only kills the triple-negative breast cancer cell lines, but not the normal cells. We can watch these cells grow and divide under the microscope. I will show you a movie now how it looks when these cells divide. Please look at the cells I numbered. They will divide as soon as I start the movie. I recorded this movie over 3 hours. Let's play it again so you can see how it looks when these cells divide. And these cells have a lot of MYC. So we found if we interfere with the process of cell division, the cells will die. I will show you now how that looks. I treated the cells in a special way that interferes with cell division. And that's how the cells look when they die. They round up, they don't divide anymore, and eventually die. As I told you, the treatment should not affect normal healthy cells in the body. So we can test if our treatment will also kill normal cells or if they are fine. I will first show you a movie of normal cells dividing without any treatment. You can see that these cells look much different than the cancer cells I showed you before. And now, importantly, these cells are not killed by our treatment. So they're dividing a bit slower, but they're just fine. And this is a very important finding. It is very important to understand the mechanism of how the cells die, how the treatment is working. Because if we know exactly what's going on, we can develop a very efficient and very specific treatment that is designed to kill only the cancer cells. So this is how an untreated cell looks when we stain for the microtubule and the DNA. I showed you that picture before. In green, you see the microtubules, the strings that form the mitotic spindle to pull apart the DNA to make new daughter cells. When we treat the cancer cell now, we cannot find any normal microtubules anymore. However, in normal cells we can. So that means that our treatment destroys the microtubules, and so the mitotic spindle cannot form and the cancer cell cannot divide. So we ask the question, why is that? Why are cancer cells more sensitive to their treatment than normal cells? And so we know that these cancer cells have MYC. So we watch the process of forming the mitotic spindle over time. Here we are looking at a normal cell with low MYC. It formed a pretty nice mitotic spindle after a certain time. However, after the same time, the cell with high MYC did not yet form the mitotic spindle. And this is the reason why it takes the cells with high MYC much longer to divide. They need much more time to proceed through mitosis than the normal cell with low MYC. We know now that MYC high cells have inherent mitotic defects. This is a vulnerability we can take advantage of to selectively kill MYC high triple-negative breast cancer. A drug that specifically targets the microtubules or interferes with the process of forming the mitotic spindle would specifically kill MYC high triple-negative breast cancer while sparing normal cells. Interestingly, there are already clinically approved drugs out there that do that. And so maybe patients with high MYC breast cancer will benefit the most from those drugs, and much lower doses of these drugs are needed and side effects can be minimized. But before we go into the patient to test our hypothesis or a new drug, we have to test them in the lab. And the best way to do that are with the so-called patient-derived xenografts. This is the best experimental system we have today, which is the closest to the patient. So we can take a piece of tumor from a patient and transplant it into a mouse. The tumor will grow in the mouse, and from there we can take out the tumor again, chop it up, and put it into many mice. We can then treat each mouse with a different drug, and can see which drug works the best in reducing tumor growth. So these mice authentically reflect tumor pathology, growth, metastasis, and disease outcomes. And so this model system has many advantages over cell lines in a plastic dish. For example, you don't have blood vessels in a plastic dish which are important for a tumor to grow. Also, you can study metastasis in the mice, which you cannot in the dish. So let me recap what I talked about today in this webinar. We learned that cell division is essential for every living organism, but also for cancer. I told you that breast cancer is not a single disease, but that each subtype is treated with a different therapy dependent on the expression of receptors. We went deeper into the mechanism of cell division, a process called mitosis, to understand how chemotherapy works. I gave you an understanding how cells evenly distribute their DNA with the help of microtubules, the green things I stained here for. You learned that Taxanes, a commonly used chemotherapy, targets those microtubules. I gave you an insight into what we are doing in our research lab. We found a factor called MYC to be very important for triple-negative breast cancer. I showed you that MYC high cells have inherent mitotic defects, and that we can take advantage of this vulnerability to specifically target triple-negative breast cancer. I also explained the latest state-of-the-art method to preclinically evaluate drugs before going into the clinic. So I hope I could give you an understanding of how we are working in the lab to understand cancer better and to eventually find a cure. My research wouldn't be possible without the support from many other people. I want to thank my supervisor Andrei Goga and all my colleagues from the Goga lab. If you are interested in more science talks, visit our webpage and click on As Seen on Lab TV. I also want to thank my collaborators from the Whitman lab for help with microscopy, my patient advocate Hannah Klein-Connolly for her support, and Susan G. Komen for financial support, and you for listening to this webinar. Thank you very much.